If you're looking for somewhere quiet to rest your mind tonight, you found it. This is a narration based on scientific facts inside the origins of life. No rush, no noise, just the slow, unhurried science of how living things first arrived, and how that story is still written inside every cell of your body. Whenever you're ready, we'll begin. 4.5 billion years ago, there was no life anywhere on Earth. That statement is worth sitting with for a moment. The planet existed, rotating, cooling, filling with water, but nothing breathed, nothing moved with intention, nothing grew. The oceans were acidic and warm. The sky carried very little oxygen. Lightning struck constantly across an atmosphere of methane and carbon dioxide. And yet, somewhere in that turbulence, something began. Not a spark of mystery, not a miracle, something far stranger and more interesting. Chemistry started repeating itself. Certain molecules, under the right conditions, can link together into chains. Those chains can fold. They can catalyze reactions, and under some conditions, they can even encourage more of themselves to form. It was not life, not yet, but it was the first shadow of something that would become life. The scientific name for that first living ancestor is Luca, the last universal common ancestor. Not the first organism exactly, more like the deepest common root. Every living thing on Earth today traces its lineage back to that population. Bacteria, mushrooms, pine trees, blue whales, and the cells lining your lungs. All of them share that ancient inheritance. The evidence is written in the genetic code itself. Across every kingdom of life, the same 3-letter sequences in DNA produce the same amino acids in proteins. That kind of universal agreement does not arise by coincidence. It points unmistakably to a shared origin. The early Earth was not a quiet nursery. It was violent and changeable. Meteorites brought organic molecules from space, adding to the chemical inventory. Hydrothermal vents on the ocean floor released hot, mineral-rich water that could concentrate ingredients and catalyze reactions. Shallow pools on rocky coastlines evaporated and refilled with each tide, cycling ingredients through conditions that could assemble and reassemble molecules repeatedly. Scientists do not yet agree on exactly which setting produced the first replicating chemistry, but the experiments run in laboratories around the world keep showing the same thing. It is not as hard as it once seemed. Simple building blocks link up. Chains form. Some chains influence what happens next. The door to life was opened not by one dramatic moment, but by the patient persistence of chemistry. Somewhere in that patience, a threshold was crossed. A molecule that could copy itself gained an edge over molecules that could not. And once copying began, however imperfect, something like evolution could begin. That is where we are starting tonight, at the very bottom of the story. Before eyes, before brains, before bodies, before anything we would recognize as alive. And yet, already, the logic was in place. Make a copy, survive long enough to make another. Everything that has ever lived, including you, is a continuation of that first, imperfect habit. The first cells were not like cells today. They were simpler, smaller, and probably much more fragile. A membrane, a thin film of molecules that keeps an inside separate from an outside, was one of the critical steps. Membranes made of fatty acids can form spontaneously in water. They can close into hollow spheres without any biological machinery directing them. And those spheres can grow, divide, and even absorb other molecules from their surroundings. Scientists call these structures protocells. Not quite alive, but structured in ways that echo modern cells. Inside those early enclosures, something remarkable could happen. Chemistry that was happening across open water could now happen in a concentrated space. Reactions that were too dilute to work in the open ocean had a chance to accumulate, interact, and build on each other. The membrane was not just a barrier, it was a collaborator. It made the inside different from the outside, and that difference is, in a real sense, the definition of life. One of the most surprising discoveries in the study of early life is the role of RNA. Before DNA, before proteins as we know them, RNA may have done both jobs at once. It can carry genetic information like DNA, and it can act as a catalyst like a protein. This double capability led scientists to propose what is known as the RNA world, a period in early life when RNA molecules were the primary engines of heredity and chemistry. The idea has gathered substantial experimental support over decades. RNA can self-replicate under the right conditions. It can catalyze its own assembly. Some of the most ancient molecular machinery in cells today, including the ribosome that builds every protein in your body, is made largely of RNA. The ribosome is worth pausing on for a moment. It is the molecular machine that translates genetic information into functional proteins. And it is found in every living thing: bacteria, plants, animals, fungi. Its core structure is so conserved across species that researchers sometimes describe it as a molecular fossil. Not a relic you dig from the ground, but a working machine that has been running nearly unchanged for billions of years. When your cells build new proteins right now, they use a machine that traces its blueprint back to organisms that lived long before animals, long before complex cells, long before the continents took their current shapes. Early life was almost entirely microbial, and it stayed that way for an astonishing length of time. The first clear fossil evidence of cells appears roughly 3.5 billion years ago. Complex animals do not appear in the fossil record until roughly 570 to 600 million years ago. That means microbes ruled the Earth, quietly, invisibly, and completely, for nearly 3 billion years. No one noticed because there was no one to notice. But those microbes were not passive. They were transforming the world. The most dramatic transformation they produced was oxygen. Certain ancient microbes developed the ability to use sunlight to split water molecules, releasing oxygen as a byproduct. This process is photosynthesis, and its emergence changed the atmosphere of Earth in ways that no geological force had done before. At first, the released oxygen reacted with iron dissolved in seawater, precipitating out as iron-rich minerals on the ocean floor. Those deposits still exist today as banded iron formations, layers of rust-colored rock that record the slow saturation of the early ocean. When the iron was gone, oxygen began accumulating in the atmosphere. That accumulation is called the Great Oxidation Event, and it occurred roughly 2.4 billion years ago. For many microbial lineages, oxygen was genuinely dangerous. It is chemically reactive, and those reactions can damage the delicate molecules inside cells. Many early organisms retreated to oxygen-free environments, deep sediments, mud, the interiors of thick microbial mats. Their descendants still live in similar places today, in the airless depths of swamps, in the guts of animals, in waterlogged soils. But some lineages adapted. They evolved enzymes that neutralize reactive oxygen compounds before they cause damage. And once those defenses were in place, oxygen could become something else entirely, a fuel source. Metabolizing food with oxygen releases far more energy than the older pathways that worked without it. That energy surplus opened possibilities that did not exist before. Faster movement, larger bodies, more complex chemistry. Oxygen turned from a threat into a tool, and that transition set the stage for everything that came next. It is worth holding that idea gently as we continue. A poison became a gift. A crisis became an invitation. And the organisms that survived the transition carried the new capability into every branch of life that followed, including the one that leads to you. The next great leap in the history of life was not a new kind of body. It was a new kind of cell. For most of Earth's history, all cells were prokaryotes, small, compact, with their genetic material floating freely inside, rather than enclosed in a separate nucleus. Bacteria are prokaryotes. So are archaea, a separate domain of microbial life that occupies some of the most extreme environments on the planet. Hot springs, salt lakes, deep ocean vents. Then, somewhere between 1.5 and 2 billion years ago, a new kind of cell appeared. It was larger. It had a nucleus, a membrane-bound compartment that housed and protected its DNA. And it had internal structures called organelles that carried out specific tasks. These were the first eukaryotes, and every complex organism alive today, every animal, plant, fungus, and most algae, is built from cells of this type. One of the most remarkable discoveries in modern biology is how eukaryotic cells came to be. The answer involves a partnership, or more precisely, an ancient act of absorption. A working hypothesis, now supported by compelling molecular evidence, proposes that eukaryotic cells arose when one prokaryote engulfed another. Rather than digesting it, the host cell and the absorbed cell developed a lasting relationship. The internal guest continued to function, providing chemical services to the host. Over generations, it became permanently integrated, an organelle. The mitochondria in your cells, the structures that generate the energy that powers virtually every action you take, descend from that ancient absorbed partner. Mitochondria still carry their own small genome, a remnant of their independent past. They still divide on their own schedule, separately from the rest of the cell. Their membranes bear molecular signatures that trace back to a bacterial ancestor. Every time your cells burn oxygen to produce energy, you are running a process that was originally developed by a free-living bacterium more than 1.5 billion years ago. Plants carry a second internal partner as well. Chloroplasts, the structures that capture sunlight and build sugars through photosynthesis, also derive from an ancient absorbed bacterium. The photosynthetic cyanobacteria, whose descendants eventually oxygenated the atmosphere, were the ancestors of chloroplasts. So in a very real sense, the same microbial group that transformed Earth's atmosphere later became a permanent resident inside plant cells, continuing its work in a new form. This process, endosymbiosis, the scientific term for partnerships where one organism lives inside another and the two eventually merge, was not a single event. It has happened multiple times across the history of life. Once we know to look for it, these ancient mergers reveal themselves in cellular structure, in independent genomes, in the rhythms of how organelles grow and divide. Life did not only compete, it also fused. And those fusions produced some of the most powerful biological innovations in the history of the planet. With larger, more capable cells available, the possibility of multicellular life opened up. A single-celled organism can do a great deal: find food, respond to light, divide, survive. But a group of cells that stay together and begin to specialize can do more. Some cells can handle movement, others can handle digestion, others can sense the environment. Division of labor among cells is the foundation of every complex body plan that evolution has ever produced. The oldest clear evidence of multicellular life appears in the fossil record around 600 million years ago, in a group of soft-bodied organisms known as the Ediacaran fauna. These creatures, some resembling fronds, discs, or flat mats, lived in shallow seas. they left no descendants that we can identify with certainty. What they tell us is that complex multicellular bodies were being explored, tested, and abandoned long before the explosion of forms that followed. That explosion came in the Cambrian period, beginning roughly 541 million years ago. In geological terms, it was sudden. Tens of millions of years, which sounds long, but is brief against the backdrop of Earth's full history. In that time, most of the major animal body plans that exist today appeared in the fossil record for the first time. Eyes, shells, limbs, digestive tracts with two openings, nervous systems, bilateral symmetry. The Cambrian seas were filling with creatures of astonishing variety, and the pace of biological invention was unlike anything that had come before. Scientists debate the causes. Rising oxygen levels may have supported larger, more active bodies. New ecological relationships, predation especially, may have driven rapid evolution of both attack and defense. Changing sea levels may have opened new habitats. Whatever the causes, the result was a world transformed. Life had moved from billions of years of microbial quiet into an era of visible, complex, moving animals. And the story was only beginning. Among the Cambrian animals, one group was small, soft, and easy to overlook. They were called chordates, organisms with a stiffening rod running along the back, a characteristic we recognize today as the precursor to the spine. Some of the earliest examples were little more than worm-like creatures, filtering food from the water. Yet that structural feature, the notochord, and later the vertebral column, turned out to be one of the most consequential designs in the history of animal life. Vertebrates, the animals built around a backbone, would go on to fill oceans, rivers, forests, skies, and eventually every continent on Earth. Fish appeared first, and they dominated the seas for hundreds of millions of years. The variety among early fish alone was remarkable. Armored forms, jawless filter feeders, predators with hinged jaws and reinforced skulls. Jaws, in particular, were a transformative innovation. Before jaws, feeding was limited to filtering or suction. With jaws, an animal could grasp, crush, tear, and pursue prey in entirely new ways. The appearance of jawed vertebrates changed the dynamics of marine ecosystems in ways that still echo in the biology of every living fish, bird, reptile, and mammal. Around 375 million years ago, some fish were living in shallow waters, where oxygen levels fluctuated. They had lungs, or lung-like organs, as a supplement to gills, and some of them had fins, with an internal structure that could push against the bottom. Over generations of ancestors that were slightly better at supporting their weight, slightly more able to move short distances across exposed mud or between water bodies, those fins became limbs. This transition from water to land is one of the most studied and celebrated episodes invertebrate evolution. Fossil species like Tiktaalik, discovered in the Canadian Arctic in 2004, preserve intermediate stages of this transition in astonishing detail. A fish-like body with a neck, a skull that could move independently, fins with wrist-like bones. It is a creature that looks like it belongs on a boundary, because it did. Life on land required solving a series of new problems. Water prevents desiccation, land does not. Water supports the body's weight, land requires structural support. Water carries dissolved oxygen to gills. Air requires lungs that could expand and draw gas inward. Reproduction in water can happen with eggs released freely. On land, eggs need protection from drying. The first fully terrestrial vertebrates, early reptiles, solved the desiccation problem in eggs with a waterproof shell and internal membranes that surrounded the developing embryo with its own fluid environment. That design, the amniotic egg, freed vertebrates from dependence on standing water for reproduction and opened every inland environment on the planet. Plants had arrived on land before animals. The earliest land plants, descended from green algae, appeared roughly 470 million years ago. At first they were small, without roots or leaves, clinging to damp surfaces near water. Over tens of millions of years, they developed vascular systems to move water upward through their bodies, roots to anchor and absorb, and leaves to capture more sunlight. Forests appeared for the first time around 385 million years ago. When trees fell and accumulated faster than decomposers could break them down, immense deposits of carbon formed, the coal seams that would later power the Industrial Revolution. The forests themselves changed the atmosphere, drawing down carbon dioxide and raising oxygen to levels higher than today. This rise in oxygen may have contributed to one of nature's stranger experiments: giant insects. In the Carboniferous period, with atmospheric oxygen roughly 30% compared to today's 21%, arthropods could grow far larger than modern forms. Griffin flies, ancestors of dragonflies, had wingspans approaching 70 centimeters. Millipede-like creatures measured over 2 meters in length. When oxygen levels dropped again in later periods, these giants disappeared. But for tens of millions of years, the forests of the Carboniferous were patrolled by things that would seem fantastical today. Mass extinctions punctuate the story of life, and each one reshaped what came after. 5 major extinctions are recognized in the fossil record, though smaller events occurred between them. The most severe happened roughly 252 million years ago, at the boundary between the Permian and Triassic periods. An estimated 96% of marine species and 70% of terrestrial vertebrate species disappeared. The causes likely involved massive volcanic eruptions in what is now Siberia, which released carbon dioxide and sulfur compounds that altered climate and acidified the oceans. recovery took millions of years. But life did recover. And in the open ecological space left by the mass dying, new groups expanded. Dinosaurs rose to dominance in the Triassic period. They were not simply large reptiles, they were extraordinarily diverse, some small and quick, some enormous and long-necked, some feathered and warm-blooded. They dominated terrestrial ecosystems for roughly 165 million years. When a large asteroid struck Earth 66 million years ago, leaving the Chicxulub crater in what is now the Gulf of Mexico, non-avian dinosaurs disappeared rapidly. But birds, which are the living descendants of a dinosaur lineage, survived. And small, warm-blooded mammals, which had lived in the shadows of dinosaurs for over 100 million years, were suddenly in a world with far fewer large competitors. What followed was one of the most rapid diversifications in mammal history. Within 10 to 20 million years after the extinction, mammals had diversified into bats, whales, hoofed animals, primates, rodents, and dozens of other lineages. The world that opened after catastrophe became one of the richest periods of mammalian evolution the planet had seen. Primates, the order of mammals that includes monkeys, apes, and humans, appeared roughly 55 to 65 million years ago in the warm forests that followed the end Cretaceous recovery. Early primates were small, arboreal, and probably nocturnal. Their defining features, forward-facing eyes, flexible hands, nails instead of claws, relatively large brains compared to body size, are all well suited to navigating a three-dimensional environment with precision. Grasping branches, judging distances, recognizing faces, tracking social relationships. These abilities have deep roots in primate biology, and they shaped everything that came after. The lineage that would eventually produce humans split from the lineage leading to chimpanzees and bonobos somewhere between 5 and 7 million years ago, based on molecular and fossil evidence. That split did not produce humans immediately. It produced a long series of ancestor species, collectively called hominins, that were distinct from other apes in important ways. The most visible distinction was bipedalism, walking habitually on two legs. Fossils like Australopithecus afarensis, known from a famous partial skeleton discovered in Ethiopia in 1974, show a species that walked upright but had a brain nearly 1/3 the size of modern humans. Bipedalism preceded large brains by millions of years. The spine, the pelvis, the arrangement of leg bones, these all reorganized around an upright posture long before the skull expanded to accommodate a larger thinking organ. The brain did expand dramatically in the genus Homo. Homo habilis, appearing roughly 2.4 million years ago, showed evidence of stone tool use. Homo erectus, appearing around 1.9 million years ago, had a substantially larger brain and spread out of Africa into Asia and Europe. Homo heidelbergensis, roughly 700,000 to 200,000 years ago, built shelters, made wooden spears, and showed evidence of cooperative hunting. Each step involved not just anatomy but behavior, ways of organizing life that were themselves subjects to selection. Modern humans, Homo sapiens, appear in the African fossil record roughly 300,000 years ago. Anatomically, that is recent. In the context of the full history of life, it is almost impossibly recent. If you compress the entire history of life into a single calendar year, complex animals would not appear until mid-November. Non-avian dinosaurs would go extinct on December 26th, and anatomically modern humans would arrive in the final hours of December 31st. We are a very new species, telling a very old story. And that story is not just history. It is biology running inside us right now. One of the most striking discoveries of recent decades is the extent to which all animal body plans share deep genetic similarities. The gene networks that specify head versus tail orientation, top versus bottom, and the placement of limbs along a body axis are remarkably conserved across animals. A gene called PAX6, which helps direct eye development in fruit flies, is related to the genes that direct eye development in mice and humans. When researchers experimentally activated the fly version of this gene in unusual places on a fly's body, extra eyes appeared, fly-style compound eyes, but eyes nonetheless. The same underlying genetic logic was producing them. This deep conservation of developmental genes, sometimes called deep homology, tells us something profound. The molecular toolkit for building animal bodies was largely assembled early in the history of complex animals and has been repurposed, combined, and redirected ever since. Eyes did not evolve from scratch more than 40 times in different lineages. Rather, a shared genetic foundation for eye development was present in a common ancestor, and different lineages independently elaborated on that foundation in different ways. The same principle applies to limbs, hearts, nervous systems, and many other structures. Life is not endlessly inventive at the genetic level. It is deeply conservative. And that conservation is what makes the diversity of bodies possible. Evolution also operates on behavior, not just anatomy. In social species, the ability to cooperate, communicate, deceive, remember, and predict the behavior of others can have enormous effects on survival and reproduction. Among primates, brain size correlates not just with ecological challenges, but with the complexity of social groups. The demands of tracking relationships, alliances, and histories in a large group may have driven some of the cognitive expansion that eventually produced human cognition. Language, the ability to refer to things that are absent, hypothetical, or in the future, added a new dimension to that social intelligence. With language, knowledge could accumulate across generations in ways that no other species had achieved. Human evolution did not happen in a single lineage. Genetic evidence shows that modern humans interbred with at least two other hominin groups, Neanderthals and Denisovans, after leaving Africa. Most people of non-African ancestry carry almost 1 to 4% Neanderthal DNA. Some of those inherited variants appear to have offered real benefits, affecting immune responses, cold tolerance, and other traits. The story of human origins is not a single clean line. It is a web of populations that met, mixed, competed, and sometimes merged. Recent evolutionary pressures on human populations have left traces in the genome that can be read today. In regions with intense ultraviolet radiation, darker skin pigmentation offered protection against DNA damage, a trait that selection reinforced over generations. At high altitudes, some populations develop genetic changes that alter how their blood carries oxygen, supporting daily life where air is thin. Populations with long agricultural histories often show higher copy numbers of the genes involved in starch digestion. And lactase persistence, the ability to digest milk sugar into adulthood, evolved independently in several pastoral populations, likely because dairy was a reliable calorie source even when crops failed. These are not ancient curiosities. They are recent adaptations, shaped by pressures that human ancestors faced within the last few thousand years. Evolution did not stop when culture began. Culture became part of the environment, and the genome kept responding. And evolution has not stopped now. Every generation, mutations arise. Every generation, some variants spread because they improve survival or reproduction slightly. In the current environment, with the current pressures. Cities, antibiotics, global travel, changing diets, new pathogens. All of these are evolutionary pressures. They are gentle, slow, and mostly invisible on a human timescale. But across thousands of generations, they will have shaped the biology of whoever comes after us, just as millions of years of pressure shape the biology we carry now. There is something quietly extraordinary in that thought. The same process that built eyes, moved fish onto land, produced flowers, and eventually produced minds capable of wondering about all of this, is still running. It has never paused. It runs through every birth, every death, every subtle difference in who survives and who does not. The story of life is not in the past. It is the present moment of every living thing. The living world holds more than 4 billion years of accumulated answers. Every species alive today is a solution. Imperfect, ongoing, but a solution to the problem of surviving in a changing world. Coral reefs harbour more species per square metre than almost any other environment on Earth. And that density of life reflects millions of years of intricate coevolution. Each species has shaped the others through competition, predation, mutualism, and coincidence. Remove one, and ripples move through the system in ways that are difficult to predict. The word ecosystem comes from that understanding. Life does not only fill environments, it makes them. Earthworms aerate soil, making it habitable for roots. Roots stabilize slopes, reducing erosion that would otherwise carry nutrients into waterways. Bees transfer pollen between plants, allowing reproduction that neither could achieve alone. Whales, when they surface to breathe, bring nutrients from the deep toward the sunlit upper ocean where photosynthesis happens. Their movements are part of a nutrient cycle that influences the productivity of entire ocean regions. Even when a species disappears, its absence is a kind of presence. Ecosystems reorganize around what is missing. In some cases, the absence of a top predator allows prey populations to grow, which reduces vegetation, which changes soil, which changes water flow. These cascades, sometimes called trophic cascades, have been documented in wolves reintroduced to Yellowstone, in the decline of sharks in coastal waters, in the removal of sea otters from kelp forests. The lesson they carry is the same lesson woven through the entire history of life. Everything is connected, and those connections run deeper than they first appear. We are entering a period in that history when one species is changing more about the living world than any single species has changed since photosynthetic bacteria altered the atmosphere. That is not a moral claim, it is an ecological observation. Habitat fragmentation, introduced species, altered nutrient cycles, shifting temperatures. These change the selection pressures that every other species faces. Evolution will continue, but what it produces and what it loses will be shaped by conditions that have no clear precedent in the fossil record. And yet, for all its urgency, the larger story holds something steady. Life has navigated catastrophes before. It has crossed mass extinctions, poison atmospheres, and millennia of ice. Not by being unbreakable, but by being diverse, by branching, by trying, by carrying enough variation that when conditions changed, something remained. That pattern is not reassurance, it is description. But there is something in it that settles when you hold it long enough. The logic of life is persistence, not perfection, not safety, just the quiet, stubborn continuation of the experiment. Your body is part of that experiment. Every cell you carry runs on chemistry that was first assembled in a world without land, without air as we know it, without anything visible to the naked eye. The mitochondria generating your energy right now share ancestry with free-living bacteria. The ribosomes building your proteins follow a blueprint older than any fossil we have found. The proteins that repair your DNA when it is damaged, they exist because organisms that could repair damage outlived those that could not. and they pass that ability down through an unbroken chain that reaches back past all complex life, past all animals, past all eukaryotes, to the very first populations that learned to copy themselves in a mineral-rich sea. You are the current version of something very, very old. Not a finished thing, a continuation, an edge of the story still being written. Let the facts loosen now. Let them drift like sediment settling after the water stills. You do not need to hold the timeline. You do not need to remember the names. All of it, the first cell, the first breath of oxygen, the first eye-opening in a dim Cambrian sea, all of it has already happened. It already led here, to this moment, to you, resting. Your body knows what to do with quiet. It has known for a very long time. Long before minds could worry, cells were resting, repairing, renewing, following rhythms older than the mountains. You carry that rhythm. It is not something you have to find. It is already running. The thread that began in a shallow, warm sea passed through every extinction, every ice age, every migration across land and water, every generation that survived long enough to pass something forward. That thread is in your hands tonight, not as a weight, as a continuity. The world is still turning. Cells are still dividing. In the soil outside, organisms are doing what organisms have always done, finding what they need, moving toward it, resting when the light goes. You belong to that, all of it. Let your breath find its own length. Let your body sink toward what it already knows. You are held by something very old, very patient, very quiet. The story of life does not have an ending. It has only a present moment. And in this moment, you are resting. 4 billion years, and all of it arrived here, at this breath. Let your eyes stay closed. Let the thoughts drift past. Like weather moving through, you are not separate from the story. You are what the story looks like right now. The night is very still. Your body knows the way. Follow the breath down. Sleep well.